Hello and welcome to another Bio-180 Tutoring Center video. In this video we'll be talking about glycolysis. Glycolysis, you need to know a fair amount about it. The problem with this unit, with metabolism, people freak out because there's so much information and they think they need to memorize absolutely every detail. I'm here to tell you you're not. You don't need to memorize every substrate and every enzyme. You just need to know the basics about what's going on, some of the key steps, some of the key regulatory steps, and a few of the enzymes will be helpful to have memorized, but you don't necessarily need to know every single step. So to begin with, the startings of glycolysis. Glycolysis, if you're not already aware, is the process the body uses to break down sugar, specifically glucose, to produce energy. And so here's our cell, and our glucose, our sugar, enters the cell. The first step, the most important step, to make sure that the glucose doesn't leave the cell, to keep it trapped inside the cell, an enzyme called hexokinase comes along and adds a phosphate to the glucose molecule. So if we do glucose, and a phosphate is added to it by the enzyme. I'll do enzymes in dark blue. Hexokinase. And you'll want to know that and what it does. So the glucose comes into the cell and hexokinase adds a phosphate, so now it's known as glucose 6-phosphate, and it cannot leave the cell. And step 2, the glucose 6-phosphate, G6P, glucose 6-phosphate, is converted into fructose 6-phosphate. It's changed to fructose 6-phosphate by the enzyme phosphoglucose isomerase. It's an isomerase because it changes the glucose into fructose, so it changes the shape in preparation for the next step in the process. I guess I forgot to mention here, this phosphate didn't come from anywhere. ATP was hydrolyzed to ADP, and this inorganic phosphate came from the ATP. And so here we've got one ATP being consumed. And then in step 3, fructose 6-phosphate is changed into fructose 1,6-bisphosphate. That's fructose 1,6-bisphosphate. And again, another ATP is being used. ATP comes in, is hydrolyzed, ADP comes out, and now there are 2 phosphates on the number 1 and the number 6 carbon. Bisphosphate means 2 phosphates. So this, so far, is known as the energy investment phase. The energy of the glucose molecule is being increased. The potential energy is being increased by adding these phosphates. It's an investment of energy. We add the 2 phosphates and do this to these changes, these chemical reactions to the glucose molecule, so we can so the cell can then further split it in half and harness the energy from it to make pyruvate and continue the process. So we put in 2 phosphates, but you'll see that more comes out. So again, in the energy investment phase, 2 ATP have been consumed. And you'll want to remember this. What happens in the energy investment phase? The 2 ATP, where they come in, and what enzymes are involved in the process. Now this step 3 is probably the most important step in all of glycolysis. This step of changing fructose 6-phosphate into fructose 1,6-bisphosphate is done by an enzyme known as phosphofructose phosphofructokinase. Phosphofructokinase, you'll want to remember that. The reason being because if we look at say this is the enzyme phosphofructokinase, if you haven't watched the video on enzyme regulation, then I would suggest watching that first. But phosphofructokinase is controlled allosterically. So if this is the fructose 6-phosphate molecule and it's supposed to be changed into the fructose 1,6-bisphosphate, this enzyme, this is phosphofructokinase, it's controlled allosterically by ATP and ADP. ATP, if it's present in the cell, will think about it. If the ATP is present, the cell has plenty of energy. So does it need more energy? No. So the ATP will bind and turn off the enzyme. And if it turns off the enzyme, it stops glycolysis. Glycolysis is stopped by ATP allosterically controlling phosphofructokinase. But if we have ADP, if that is present, the cell needs more energy. Glucose needs to be burned. So if ADP is present in higher concentrations, it will bind to this non-active site and it will activate and turn on phosphofructokinase, and so glycolysis continues. And this is why phosphofructokinase is the most important enzyme in the whole process, and you will want to have a deep understanding of phosphofructokinase. Next, we move on to the energy payoff phase. And so the fructose 1,6-bisphosphate is split into two molecules. It's split into glyceraldehyde 3-phosphate and dihydroxyacetone. And this, I guess, would be step 4. It's split by the enzyme known as aldolase. Again, that's a name you'll want to remember. The dihydroxyacetone is then converted into G3P, so we end up with two molecules of glyceraldehyde 3-phosphate. And a simple isomerase enzyme does that. You don't need to worry about the name, but just know that the fructose 1,6-bisphosphate molecule, when it gets split, it doesn't get split perfectly. It is not a mirror. Like if we look at look down the middle of this molecule here, if you were to split it in half, which what happens, aldolase splits it in half, it doesn't, each half of the molecule is not identical. So we get the glyceraldehyde 3-phosphate. This is needed. Dihydroxyacetone is not needed, so it takes an additional step done by an isomerase to change it into another glyceraldehyde 3-phosphate. That's glyceraldehyde 3-phosphate. Now with step 5, the glyceraldehyde 3-phosphate is going to be changed into glyceraldehyde 1,3-bisphosphate. And remember, there are two molecules, and so we went from having one phosphate on each molecule to having two phosphates on each molecule. So two inorganic phosphates were consumed and added to, I guess, ignore this arrow, they're added to the molecule. And at the same time, we have a molecule known as NADH produced. So the enzyme that does this reaction is called triose phosphate dehydrogenase. Dehydrogenase, you need to know that name. Dehydrogenase do redox reactions, and in this step, the energy necessary for the inorganic phosphate to be added comes from the process of removing the electrons and the hydrogen from the molecules. And so 2 phosphates are consumed, no ATP consumed, just floating inorganic phosphates, and 2 NAD plus are reduced into 2 NADH, and this can then go off to the citric acid cycle and continue the process of cellular respiration. But knowing this name, dehydrogenase, and what happens during step 5 is important, that the phosphates are added to make G13BP, and then the NAD is reduced to NADH, and this molecule is very important for the rest of respiration. And then step 6, the glyceraldehyde 1,3-bisphosphate is split into both of them are split into molecules of 3-phosphoglycerate, and there's steps 6, 7, 8, and 9, and we go from glyceraldehyde 1,3-bisphosphate to 3-phosphoglycerate to 2-phosphoglycerate to phosphoanyl pyruvate to pyruvate. Again, there are 2 molecules each time. And different enzymes do these different processes, and we don't need to worry too much about those. But during these last few steps, several molecules are produced. NADH has been produced, but then during these steps, 4 ATP are made through substrate level phosphorylation. 2 H2O molecules are given off, and of course 2-pyruvate are the final product of glycolysis is pyruvate. So this is the energy payoff phase. Energy payoff phase. And in this process, 2 NADH were made, 4 ATP, 2 H2O, and 2 pyruvate. These are the products of glycolysis, and you want to remember that, especially this. We've got a net gain of 2, a total gain of 4 ATP, but because 2 ATP were consumed already, there's a net gain of 2 ATP for the whole glycolysis. That's why it's called the investment phase. Energy is put into the molecule to raise its potential energy, and we use 2 ATP. And then, like all investments, you're wanting a payoff, you're wanting a profit, and the profit is right here. And the pyruvate and the NADH, they're all going to pyruvate, NADH, they're going to go into the mitochondria to produce more energy, and you'll learn more about that later on. But that's why it's the payoff phase, because we gained more energy from the glucose than we originally added. This is a difficult process to grasp for lots of students. I would suggest drawing it out several times. Draw it out, try color coding somewhat like what I did to help you visualize it and help you memorize it. But again, don't focus on learning names like here. I just glanced through the material. You don't need to know those names, the enzymes that do them. You just need to know what goes in. So the glucose comes in. Some enzymes change its structure to increase its energy. ATP is consumed, and then these molecules, I'm not going to say them again, are given off as profit, you could say. Just remember hexokinase, phosphofructokinase, aldolase, and dehydrogenase. Those enzymes are the most important ones to remember. I hope this video helped. Thanks for watching, and good luck.